Hi, yeah, so I'm Kat, and I'll be talking about bioinformatics in medical research. So just out of interest, how many people here have heard of bioinformatics? Oh, two people from the audience. That's more than what I expected. So I'll come on to what bioinformatics is in a minute. So I actually did my PhD at Newcastle University, and that was funded by CIMA. And that was part of the first cohort of students that were trained by CIMA. And then I stayed on afterwards to work as a bioinformatician. So really, I've got CIMA to thank for my PhD and also now my job. So before I go on to say what bioinformatics is, I think it's important to introduce DNA sequencing and how we use this in research. So some of you may remember from your GCSEs or O-levels that DNA is made out of 4 bases: adenine, thymine, guanine, and cytosine. And the whole set of these 4 bases of the DNA in a human makes up the genome. And by looking at the genome, we can find, which is basically the blueprint of how to make a person, that particular person, we can look at the sequence of their genes and find out whether they've got a mutation, for example, that might infer risk to a disease. And by comparing sort of patients with a disease and patients without a disease, we can then say, like, well, it's because this T has turned into a G, and that's probably why this person has more of a risk of developing osteoarthritis, for example. So apart from sequencing the DNA, we can also sequence the RNA, which is a related technology of RNA-seq to determine which genes are switched on and switched off, because of course not every single gene is switched on all the time. And we can also find out how these genes are regulated, so how these genes are switched on and switched off by smoking or obesity or diet. So the first time the human genome was sequenced was in 2003. Well, that's when the Human Genome Project was completed, and actually it was sequenced over, I think, over 15 years. So it was a massive effort, and it took many scientists across many different countries in order to do this. And since then, we've come a long way. We can now sequence a human genome in a matter of days. And in 2012, the UK government announced the 100,000 Genomes project, which aims to sequence NHS patients with common cancers, rare diseases, and their families, if possible. And the idea behind this is that researchers can then mine this information to find out why some patients get diseases and why others don't, and how these diseases are inherited. So within Newcastle University and in projects funded by CIMA, we also use DNA sequencing and related technologies in our projects. So for example, we extract DNA from osteoarthritic patients, that you've just heard from Joanna. We can then compare them to healthy patients to find out which genes that the osteoarthritic patients have that the healthy patients don't. We can also ask questions such as, is osteoarthritis the same depending on which joint site it's in? So is hip osteoarthritis the same as knee osteoarthritis, for example? So these are all studies that are currently ongoing at the moment to try and understand the disease process. So I think most of you here will have been exposed to DNA sequencing through the media. So DNA sequencing in the news, so these are all headlines that are found on the BBC News website, not the Daily Mail. So how knowing your genetic code could lengthen your life, how we can use DNA sequencing to study evolution, how we can use it to find out whether you're at risk of a certain cancer, for example. So what you're less likely to know is how is this done? Like, how do researchers get this information and how do they come to these conclusions? So Joanna's already explained how DNA is extracted from samples, so I won't go into that. But once the DNA is extracted, it goes into this huge machine called a DNA sequencer. And eventually, this machine will spit out a whole load of information in the form of these 4 letters, A, T, C, and G. 3 billion of them in the human genome. So typed out in its entirety, this would fill 200 telephone directories. So as you can imagine, it's basically impossible to go through this by eye and to sort of make any sense of it at all. So how do researchers do that? Well, that's where bioinformatics comes in. So bioinformatics is a relatively new and little-known field, at the intersection of statistics, computer science and biology. And bioinformaticians will receive training in all three disciplines. And we basically analyse all the data that comes out of a machine to try and make sense of it, to try and sort of find mutations, to find differences and patterns and trends that a diseased patient may have that a healthy patient doesn't, for example. So we run analyses and algorithms, mostly on a computer, and that's where the computer science comes in. We use statistics to test whether patients with a disease really do have this mutation or not compared to healthy. And we do many other things, which is why it's quite difficult to define what a bioinformatician is. And of course, we also aid other biologists and researchers in interpreting these results. And in many cases, bioinformaticians are also biologists themselves. So I come from a biology background. I did my undergrad degree in biology before moving into bioinformatics. So whilst all of this might sound a bit boring, a bit dry, sort of discussing algorithms and data, I think it's quite important to know where patient data and potentially your own data is and what happens to it. So there's a huge increase in data generation over the past 10, 15 years, and bioinformatics is a rapidly growing field. Currently, there's a huge shortage of bioinformaticians in the UK and actually across Europe and the world because of this huge increase in data generation that's happened fairly recently. And of course, because of this huge increase in data, we need to ask the question what to do with it. How is this data stored? Are there any security concerns? And if you as patients have your genome sequenced as part of a research project, who owns your genome? What happens to your data and who has access to it? Is it ethically used? So I think these are all questions that we need to consider in the light of this whole sort of data revolution. So I think in the future this type of research will be increasingly common, and computers have taken over nearly all aspects of our lives. Medical research is no different. I think in the future, biologists are just as likely to be working on a computer as in the lab. And with the development of new technologies and the rise of artificial intelligence, it could be that in the future, computers or robots could be helping us to diagnose and treat diseases. So this kind of field of research has come a long way since the structure of DNA was discovered in 1950 and in the sort of past 18 years or so since the human genome has been sequenced. And that's all I want to say.